I'm Shrija Das. Today, I'm going to focus on the chapter The Cell. That is the chapter 8 of the book. So, we are going to focus the chapter The Cell. Now, let us see the slides which I have prepared for you. Now, the cell which is actually the structural and functional unit of life, is derived from a Latin term which means a little room. So it was first discovered by Robert Hooke in the year 1665. He obtained these dead cork cells and he saw it under the microscope, his self-designed microscope, and saw hexagonal type of structures, and he named them as cells. So cell has been derived from the term cellular, which means a little room. Now, how the cell is seen? So we have already, I have already told that Robert Hooke, he saw this under a self-designed microscope which he designed. Let us come to the point, that is the microscopes. So what is a microscope? So microscope is an instrument that makes us able to see the enlarged image of an object which we cannot see by our naked eyes. Okay, so there are various types of microscope: simple microscope, compound microscope, electron microscope, and again there are various types of electron microscope even. So simple microscope, it involves only one type of lens, convex lens or converging lens. So here magnification is less, a little less as compared to the compound microscope, because compound microscope is a type of microscope which involves two types of cells. One is objective and another one is eyepiece. So both two converging lens are used in this compound microscope. Now, in electron microscope, a beam of electron is used to see the microscopic structure which are found inside the cell. That means this electron microscope helps in viewing the structure, the internal structure of the nucleus. What are the components which are present inside the nucleus? So we can see the cell structure of the cell, enlarged image of the structure of an object, microscopic object, by the help of compound microscope. And if we want to see the further the small organelles and small components of the object, we can use this electron microscope. But this compound microscope we are using in the lab, so you can handle this compound microscope after getting instructions from your teachers or lab demonstrators. But electron microscope are used by experts, the research workers, the scientists. Okay, so now let us see the structure of the compound microscope. Now you can see the structure of the compound microscope. It has an eyepiece and an objective, as you can see. So this eyepiece and objective are two convex lens which are present and which helps in the proper enlargement of the image, enlargement of the object in the form of image, which is obtained in the slide. So this one is the eyepiece, this is the body tube, this is the coarse adjustment which you can adjust so that to reach the proper image. Here is the fine adjustment. You can finely adjust until you get a very fine structure of the cell or any object you are viewing. This is the arm. So you should always hold the compound microscope with the help of the arm, and you should not hold it anywhere else. This is the stage on which the microscope is kept, and the clips are put on the this microscopic slide. This one is a slide containing the object. Okay, so this slide, it can be seen, the object can be seen on the slide without any disturbance. So for that we are using these clips. Then this is the stage on which the slide is being placed. Here is the base and this one is the mirror. So, the compound microscope is a light microscope. That means it uses the sun rays or sunlight in order to reflect into this slide. Okay. Now, coming to this, let us see. Now, let us see what is the difference between a simple microscope and compound microscope. In a simple microscope, we are, as I told you, we are using only one type of convex lens. But in compound microscope, we are using about two type of convex lens, that is one eyepiece and objective. So eyepiece lies close to the eye and objective lies close to the object. Like we can define it like this. So in this case, object is placed within the focal length of the convex lens. But in compound microscope, the object is placed away from the focal length. That means beyond the focal length of the objective lens. Now the magnifying power, as you know, it is less as compared to the, as I've discussed, the compound microscope. So this has compound microscope, otherwise called as light microscope, has more magnifying power as compared to the simple microscope. Now here are the examples. So we can observe minute parts of a watch to read words in small prints, etc. But here you can observe the blood corpuscles or the blood cells, the plant cells, the animal cells, etc. So you can easily differentiate between the simple microscope and compound microscope. Now let us first see what are the types of organisms. Okay, so these are the types of organisms which you differentiate based on the number of cells present in the organism, like unicellular organisms and multicellular organisms. As uni means one and cell. So one cell organism, here many cell organism, multi is many. So unicellular organism consists of one cell. Inside it, all the biological process takes place, like digestion, respiration, excretion. Within one cell, everything occurs. But in multicellular organisms, so complex, so cells, many cells are present there, a complex structure of cells are present there. So each and every cell, they perform different tasks by forming tissues, then by forming organs, organ systems. Okay. Along with that, the single cell also perform the basic functions, and the cells together also they perform basic functions of life. Now these organisms, they are examples are amoeba, bacteria, paramecium. Multicellular organisms, like you all know, these are plants, animals, okay, these are human beings. So next, let us come to the cell theory. Cell theory was first formulated by M.J. Sleden and Theodor Swan. So both of them were German biologists. So first, M.J. Sleden, he was a scientist who, in the year 1838, who found some observation after studying the plant cells. Whereas this Theodor Swan, okay, so he studied on the animal cells in the year, the very next year, that is in 1839. So the main principles of the theories are, they postulated, so those observations they postulated under one theory, that is all living organisms are composed of cells, and the cell is the basic unit of life, that is basic structural and functional unit of life. Now, this cell theory was further, it was modified by another scientist called as Rudolf Virchow in the year 1855. So, according to him, cells arise from pre-existing cells. So, in Latin terms, he expressed that is omnis cellula e cellula. Sometimes questions may come that who gave the term this omnis cellula e cellula, in which year, and what is the meaning of this term, so you can answer it properly. Now let us come to one by one. Let us come to these all the theories which have been the postulates of the theories, that all living beings are composed of cell. Like a paramecium is an example of a single cell. Monkey is an example of multicellular organism. A cactus is an example of even multicellular organism. That means this organism is made up of single cell. This organism is made up of many cells, and this also it is made up of many cells. That means everybody's body are made up of cells. Okay, so maybe it is a one cell organism, maybe it is multicellular organisms. Now, cell is a basic unit of life. That cell performs all the life functions, and also as well as it makes the total structure of the organism. Like here it has been given, it performs all life functions like intake of food, ingestion, digestion, metabolism, excretion, respiration, your circulation, irritability, so etc. Irritability means the response to the stimuli. Now, you can see that each and every cell, a single cell, the division of labor occurs. That means the functional aspects are divided between the organelles. Like the mitochondria, they are the powerhouse of the cell. They generate ATP in the energy in the form of ATP. So this energy is used by the cell for its performing all the activities. Then other organelles like your ribosomes, they are the site for protein synthesis. Okay, now all these are very, very important, these organelles which carry out the physiological process inside one cell. So each cell organelle performs different functions like we have already discussed: clearing the waste material, protein synthesis, lipid synthesis. Okay, like now next is cell arise from pre-existing cells. Cells arise from pre-existing cells. Like a prokaryotic cells splits into two identical cells by the process of binary fission. Already in standard 7th you have learned what is a cell fission and what do you mean by binary fission. Like in this, see, a cell divides into two, okay, into two cells of same size and having the same equal genetic material, similar genetic material. So, for example, here a eubacteria has been given. Now, children, we will talk about this prokaryotic and eukaryotic cell in my next slide. So, prokaryotic means pro means primitive and karyon means nucleus. So, these organisms have a primitive type of nucleus, and eukaryote means modern type of nucleus. That means a true nucleus is present, having definite nuclear membrane. Okay. So in case of prokaryote, they reproduce by asexual methods, that is a binary fission. But in case of eukaryote, mostly the eukaryote are composed of somatic cells which make up their body structure. So they undergo mitosis, in which again the cell splits into two identical daughter cells and this process is called as mitosis. And again another type of cell is also present called as a germ cells, which help in reproduction, but they undergo meiosis, okay, which helps in sexual reproduction. So that we are these are so from this we can conclude that cells arise from pre-existing cells. They have come from no other source, rather they have come from pre-existing cells. Now next is the level of organization. So how the level of organization occurs in a cell and in multicellular organisms. Let us see. First of all, this is the atomic level, in which the atoms they are like your carbon, hydrogen, oxygen, so having a different definite property. So these atoms they are constitute the atomic level. And then the molecular level, so these atoms like carbon, hydrogen, oxygen, they combine to form molecules which have a totally different property as that of the atomic, in the substances which are in atomic level, like that of your carbohydrates like glucose. So glucose here is a molecule which is a macromolecule. So this glucose consists of carbon, hydrogen and oxygen. So here in glucose, the property of the glucose is different as of the carbon, hydrogen and oxygen. Okay, so in molecular level, these are some of the examples. So these components are formed. Next, these components are surrounded by a membrane, okay, and this is called as a cell, a living cell, where all the components, the cell enclosed by a membrane or cell wall, and they perform the specific functions. How they perform? Because all these components help in what? Preparing or taking carrying out the basic functions. Like the cells have organelles inside it, and these organelles help in the proper functioning of the cells. Like example, muscle cells, skin cells, neurons. So they have specific functions. Okay. Now these cells, so number of cells together in this perform a specific task are called as tissues. And in the tissue level, examples are muscles, like epithelial muscles, epithelial tissue, connective tissues. Like connective tissue means like your blood, bone, cartilage. So those connect different parts of the body. Okay. Now so tissues, they combine to form organs. So an organ like heart, kidney, liver, stomach, okay, lungs. So all are organs, and they what carry out a specific task or specific function they have, and these tissues, they work together to complete a specific task in the form of organ. Now, various organs, they form an organ system. So here you can see an organ system like your digestive system, circulatory system, involving various types of organs. Like you can see here, the levels of organization. These are, suppose in the heart, we're talking that these are cardiac muscle cells. Then it forms tissues. Then in case of it forms the organ, that is the heart. And in circulatory system, you have the heart, you have the blood vessels, arteries, veins, capillaries. And again, the circulatory system, it involves lungs, which is another organ. Okay. Now these are the levels of the organization. Let us come to the shape and size of the cell. So there are various shape and size of the cell. So variety of types of shapes and size of the cells are present in human body. When it comes to the human body, we have different shape and size of the cells. And why it is there? Because based on their specific functions, they acquire this type of shapes. Like the nerve cells, they are elongated, you can see here. And this nerve cells, they are the longest cells, about 90 cm. Okay. And what happens actually? So this nerve cells, since it carries the information from, or helps in the transmission of the signals, okay, impulses. So since then, their elongated structure. Then you can see this one is the sperm cell. So sperm cell has a long tail, so that it can easily travel through the reproductive tract of the female. So it helps in the movement of the sperm, easy movement. Therefore, it makes it active. Now here ovum, the structure is like that, and here even the blood cells, so they are biconcave, so which helps, which can easily flow through the blood vessels. Okay. Then these are the fat cells. So it has a larger vacuole so that it can store fat in it. Even the smooth muscle cells. So all these have specific functions. So depending on the specific functions, they have different structures. So you can see here the smallest cell. You remember, children, the smallest cell is mycoplasma, which is pleuropneumonia-like organism. So it resembles that of the pneumonia bacteria. Therefore, it is named as pleuropneumonia-like organisms. So it is about this. So children, first we should know that how the cell is measured. What is the unit of measuring? So the unit of measuring is micrometer. So 1 micrometer is equal to 10 to the power minus 6 meter. So as we can write here, 1 by 10 to the power 6 meter. Okay. Now the largest cell is the egg cell of ostrich. Okay. Now this is 15 cm, about 15 cm now. But some cells are there which can change their shape. Like for example, your amoeba, which is a unicellular organism. So you know that amoeba has an irregular shape and size. Why? Because they undergo nutrition process by the help of the streaming movement of the cell. So what is the streaming movement? That means the cytoplasmic movement of the cell, because they throw their false seed or pseudopodia in order to engulf the food particles. So this food particles, it takes into and forms a food vacuole. So by this it changes its shape to reach the food. Therefore, it has an irregular shape since it undergoes phagocytosis, right? Phagocytosis nutrition. Now, this our in our body we have also some cells which have changing shapes, like your WBC type of WBC called as macrophages, and these macrophages, they also are phagocytic in nature since they have a tendency to engulf the foreign bacteria or foreign agents which are coming into our body, as they are helpful for protecting our body from the foreign invaders or foreign antigens or your bacteria, viruses which are entering into our body. So they change their shape like that of amoeba and they engulf the foreign body to protect our body or makes our body immune. Right? So next slide, it deals with the different kinds of plant cell, and again the plant cells have even different kinds of cells like that of the onion cells. You can see here the onion epidermal cells, so they are flat in structure having a nucleus. Now why they're flat in structure? Because the epidermal cells are the outermost cells of the organism, so they need to be very flat and elongated so that they can easily cover the upper surface of the body. So therefore, depending on the function, specific function, the structure is like this. Now coming to the guard cell, as you know that this guard cell are helpful for the opening and closing of the stomata, for the diffusion of gases and as well as water vapor into and out of the cell. So these guard cells, these are bean shaped cells, okay, and sometimes it is they are also dumbbell shaped. So in here they are bean shaped cells having chloroplast in them, which contains a green pigment cell wall. So these cells, they are very flexible, and why it is made flexible? Because when they will absorb water, they will swell up, and when they will swell up, it will tend to go far away from each other and causing the opening of the stomata for the diffusion of the gases and water vapor. And when the water scarcity will occur, then these will get shrunken. As a result, they will close the stomata, which will prevent the excess of transpiration or excess of loss of water from the pores. Now coming to the root hair cell, you know these root hair cells are present in the roots, small hair-like structure, and you can see they are needle-like structure, much elongated-like structure, so that they can penetrate deep into the soil and they can absorb the water from the soil. They can increase the surface area for the absorption of water from the soil. So these root hair have specific function, therefore the structure. So you can see these all belong to the plant cells, but they have varying structures depending on the specific functions they are undergoing. Right. Now coming to the next slide, there are some questions. So like dash are the basic unit of our body, and amoeba and paramecium are examples of the organisms. So let me answer all the questions. I think you all are able to answer these questions. Like dash are the basic unit of life. Cells are the basic unit of our body. The amoeba and paramecium are examples of unicellular organisms, a group of similar cells that carries out a particular function forms a, what is this? Now this one is the tissue. Who first identified cells and when? Robert Hooke first identified cells in the year 1665. And what is cell theory? We have already discussed. Write the postulates of the cell theory. Now this one is the answer which I have already discussed. This is the cell theory, and this is a very important question which usually come in exams. Okay? Now let us come to the next part, like the structural organization of a cell. Now let us see what actually a cell contains. So what are the structures, what are the substances that a cell contains? So cell usually contain a plasma membrane, okay, like you can see here. It is an example of animal cell. You can see here it consists of a plasma membrane. It is the outermost one in animal cell. Now it consists of plasma membrane, nucleus, cytoplasm, cell wall in only in the plant cell, which is absent in animal cell. So that is the main difference between a plant cell and animal cell, children. You just remember it. Now, and the cell organelles. So here there is a total concept map of a cell, or the total flow chart, the flow chart of a cell. So what are the components? Main components are the cell contains cell membrane, cytoplasm, nucleus. So this is I'm talking in general, okay, not specifying that whether it is an animal cell or plant cell, in general. So cell membrane, cytoplasm, nucleus. So cell membrane, cytoplasm, nucleus are the major thing, but only one I want to add that I've written it here, that plant cell, they have a cell wall, an additional part, that is cell wall. Now cell, in the cytoplasm of cells, we have cell organelles as well as some cell inclusions. Okay, so cell inclusions means some of the granules are present in the cytoplasm, floating in the cytoplasm. These are the nutrient granules. So these are the granules and the vacuoles. And the cell organelles involves endoplasmic reticulum, the mitochondria, the Golgi apparatus, the ribosomes, the lysosomes, the centrosomes, and the plastids. And these plastids are found in only in the plant cell, not in the animal cells. Now here coming to the nucleus again, it consists of a nuclear membrane, which is a porous structure, double membrane porous structure. And I'll discuss why it is porous in structure, as it allows the movement of different substances, okay, in and out of the nucleus. Now nucleoplasm, this is the liquid which is present inside the nucleus. The nucleolus, this is again the substance which is present inside the nucleus. We'll discuss it. Then chromatin fiber, there's a genetic material which is present in the nucleus. As you know, the nucleus is a controlling agent of the cell. So it will control when the cell will divide and when the cell will grow. So they have the chromatin fibers. So this chromatin fibers, they are passed on to generation to maintain the, during the reproduction process. This are hereditary material which is carried on from generation to generation to maintain the species of the organism. Now, let us come to an animal cell. You can see here, the animal cell consists of a outer cell membrane, and the cell membrane is very flexible in nature, as they are made up of phospholipids. So they are flexible, they are made up of phospholipids, and also some proteins are embedded into it. Then this one is the cytoplasm part. Actually, this total is protoplasm. Nucleus plus cytoplasm is the protoplasm, which is the living component of the cell, and this one is the cytoplasm. Okay, you can see here. Here it has been written as cytoplasm. Now this one is the nucleus. Okay, this total one is the nucleus containing the. You can see that this nucleus contains the nucleoplasm. So this inner material, the liquid present inside the nucleus, is the nucleoplasm. Okay. Now this one is the nucleolus, and this nucleolus plays an important role, because this nucleolus helps in the biogenesis of RNA, okay, RNA synthesis. Now here comes the mitochondria, which generates ATP or provides energy to the cell in the form of ATP. These are the ribosomes, which are the site of protein synthesis. Okay. Now you can see here, these are the endoplasmic reticulum, which are very close to the nuclear membrane. Okay. So again, endoplasmic reticulum, they are again, we will discuss that two types based on their presence and absence of ribosomes. Like rough endoplasmic reticulum, they have rough endoplasmic and smooth endoplasmic. So rough endoplasmic reticulum have ribosomes, whereas smooth endoplasmic reticulum do not have ribosomes. Then the Golgi apparatus. Then these are the centrioles which help during the cell division. lysosomes, which are also called as suicidal bag of the cell. So they help in the degradation of all the residue substances or the toxic substances present in the cell, and also they are capable of digesting the total cell. Okay, so they have hydrolytic enzymes. So this is the total structure of the animal cell. Now you can see the structure of a plant cell. The plant cell consists of a cell wall. You can see this one is the cell wall. It's a rigid structure made up of cellulose, hemicellulose, okay, pectin. So all these are called, this one is the cell wall. And inner to that is a cell membrane. So whereas in animal cell, the cell wall is absent and the cell membrane is present. Now again, everything is, all the organelles are present like Golgi apparatus and endoplasmic reticulum. Okay, ribosomes all are present, microbodies are present, ribosomes are present, but difference is that it has a large vacuole. Okay, it is a large vacuole and the nucleus is pushed to the periphery of the cell or the to a side of the cell, because vacuole is very large for the storage of different substances. Okay, storage of water, minerals, different substances. Now this one is the nucleus. Again, it has smooth and attached to smooth and rough endoplasmic reticulum. So this is, and the most important feature is that this plant cell have plastids. So these plastids are of many types. So this one of the plastids has been given. This is the chloroplast, and this is the green pigment, which chloroplast containing the green pigment, that is the chlorophyll, which help in the photosynthesis process, that is preparing its own food. Now you can see the difference between a plant cell and animal cell, as we have discussed before, that one major difference between the plant cell and animal cell is the plant cell, they have cell wall, animal cell, they do not have a cell wall, and the plastids are present in plant cells, whereas in animal cells, the plastids are not present, they are absent. And here the nucleus in animal cells, they are present in the center of the cell, and they are very well visible, whereas in plant cells the nucleus are pushed to the periphery, and to the center they contain large vacuoles for the storage purpose. Now this the chloroplast, but other these things, they are same in that of the plant and animal cells. Now this is the process of cell multiplication. So how the cell multiplies, already I have discussed at mitosis. You can see the total division of the cell. So why the cell divides? Because the inner material of the cell increases, that is the DNA replication. So when it increases, the cell tends to divide, and here this is the, in mitosis, the cell divides into two daughter cells having same equal, so same in size and equal genetic material. So these are two daughter cells. So this one is a cell division, and it is very important. Okay, so now I want to wind up this chapter. So thank you so much.